session is I'm going to talk about some of the updates taking place in CMT type 1B and CMT-X. And I'll be happy to answer questions at the end as time permits. So one of the big efforts that we try to do with CMT 1X and CMT 1B is to try to get to be clinical trial ready. And that's just another way of saying that if we have candidate treatments and we want to do clinical trials, we have to make sure that we're able to interpret the clinical trials. And so I'm going to talk briefly about how we evaluate patients at our clinics within the Inherited Neuropathy Consortium to use clinical outcome assessments in both what are called cross-sectional and longitudinal studies, what we're doing with biomarkers or biological markers of the disease that we can connect with these clinical outcome assessments. And then I'm going to talk about what's going on with these outcomes and biomarkers in current studies that are actually supported by the CMTA and STAR, and briefly mention treatments on the horizon, although I think that's going to be really the topic of another session. So what I first want to do is just introduce very briefly CMT1B and CMT-X. So this is a cartoon of a protein that's in the myelin of peripheral nerve, and it helps wraps of myelin stick together so that myelin can form normally. And it's called myelin protein 0 or P0. And you can see this is the protein sequence of myelin protein 0. And this is in between adjacent myelin wraps, so when they're wrapping on top of each other. And this is on the inside of the myelin, and it crosses the myelin membrane one time. And so it's what we call a single pass protein, and it has 250 different amino acids in it. And mutations in over 200 of them can cause CMT1B. And there's a little bit of a naming issue here because some of these mutations cause early onset disease, which affect infants and children. But other mutations in the same gene don't affect people until adulthood. And sometimes they get classified as CMT-I or CMT2I or CMT2J because they damage axons a lot. But all of these are caused by mutations in the myelin protein 0 gene, so I just call them CMT1B for simplicity. And again, mutations in this MPZ gene cause CMT1B. Now, this is another protein that's in the peripheral nerve myelin, and this protein is called connexin 32, and you can see its amino acid sequence here, and there are over 400 different mutations in connexin 32 that cause CMT, and they cause CMT-X, which is actually the second most frequent cause of CMT that's known. To make it a little confusing, the gene is not called connexin 32, it's called gap junction beta 1. The protein's called connexin 32. But this is the cause of the main form of CMT-X. Now, when we have patients and we're trying to develop treatments for CMT1B or CMT-X, what we want to do is develop ways where we can meticulously and accurately measure the neuropathy so we can tell if it's getting worse or getting better. And we use these as what are called clinical outcome assessments, or COAs. And we've developed these over the last 12 and 13 years in the Inherited Neuropathy Consortium. And we've developed them for adults and more recently for children and even infants. Now, these are ways to look at CMT clinically, but we also want to look in ways that are not invasive, but in patients themselves to look as biological markers to show what's happening with the disease. And these are when we look at blood and skin and even imaging, and I'll show you a little bit of those as we go forward. So hopefully that's making some sense. I'm going to begin by just briefly reviewing our clinical assessments. We use composite scales, which are ones that we can do quickly in clinic and are based on questions that we ask you about with the CMT and findings on the exam. And we use techniques that are statistical techniques called Rasch modified scores to make sure we can distinguish small changes even in mild or severe patients. Then we also developed functional scales where we have people do something like the 6-minute walk or getting up from a chair and getting down, trying to balance, trying to use pegs and look at dexterity in the hands. And we've done this for CMT Peds, what's called CMT Farm, and the infant scale. And then finally, on these clinical outcome assessments, we've recognized over the years that there's a danger in having doctors design and use clinical outcome assessments because it doesn't get enough of the input from patients who have the disease. So we've developed what are called patient-reported outcome measures. And in adults, one is called the CMT Health Index. And in infants, in children, there's a CMT Pediatric Quality of Life instrument we've also developed. And I don't have time to spend detail on these, but I'm going to go through them very quickly. So first of all, in order to be able to make sense of these measurements, we need to have lots of patients because there can be variability from one patient to another. So what we've done is use the Inherited Neuropathy Consortium, which has been supported by the CMTA as well as the MDA and the NIH for over 12 years. And these are just where some of the sites are, and you can see they range from Australia to the United States. And the main outcome measure we've used over the years is called the CMT Neuropathy Score, which looks at, in yellow, symptoms people have, in green, findings on the neurology exam, and then some changes on nerve conductions. We've also learned with a lot of input from patients that nobody likes the EMGs, and they also can be expensive. So for many of the studies, we've dropped the EMG component of this and just used the yellow and green, and we call that the CMT exam score. This is the score that we've been using since 2009. So in a lot of our longer longitudinal studies, we use this as the main measure, the CMT exam score. But we have developed functional scores, which we are increasingly using along the way. This is the CMT pediatric scale, and you can see the dexterity, balance, ability to jump, ability to measure strength very precisely. And this is a version in kids called the CMT pediatric scale. And we've extended this into adulthood. This is similar to the pediatric scale, but it has some differences to make it a little safer for adults who have balance problems. And this is called the CMT Farm. And we've extended this, as I mentioned, even into infants, so we can measure clinical impairment from age, from birth up to early childhood with this scale. So putting this all together, we now have clinical outcome assessments that we've looked at over the life of individuals. Now, this is about CMT 1B and CMT-X, and so I'm going to show you how we've done this. I'm sorry, this is just to show you the patient-reported outcome. So what we first have done is, as we've recruited patients over the years, we look at the patients that we recruited at baseline visits, and then we've used the CMT exam score to correlate how severe the neuropathy is and compare it with age, figuring that as people get older, they get more affected. As we then accrue data over the years, we can extend this into longitudinal data, which we've done for CMT-1B, and this is work that's being submitted for publication within the next month, in which we can show that using that CMT exam score, we can detect clinically significant change after 2 years with this that you can measure and we could use to determine whether there's a slowing of progression. These are with our composite scores. We've also done the same thing with CMT-X. So we've compared the baseline scores in patients with CMT-X to age. And this is work led by Dr. Scher and his colleagues. And we can show, again, there's progression. There's a significant difference in severity in younger versus older patients, men. And we also can show that the scores tend to stabilize in women with this outcome. But again, these are cross-sectional studies. And so we have extended these into true longitudinal studies where we look at how the same patients progress from year to year. And we have this work almost ready to be submitted. And this is being led by our London group, and this will be submitted by the end of the year. But again, what I've talked about is what we've done with our early exam scores, like the CMT exam score. But the more recent outcome measures that I talked about, like the FARM, the infant scale, the pediatric scale, and the patient reported outcome instruments, are ones that we've only developed and published within the last 2 years. So what I'll talk about ultimately is what we're doing now to look at those outcome measures and to see if they are more sensitive measures to detect change in CMT1B and CMT-X. So those are our clinical outcome measures. And now I want to talk a little bit about the biomarkers that we're looking at in CMT1B, CMT-X, and other forms of CMT, because we want to be able to compare these findings, which reflect the biology of the disease, with the clinical findings. And some of these biomarkers can show if the treatments ultimately are responsive to the biological problems, in which they would become normal with effective treatment. If they can show us that treatments are actually hitting the appropriate target, so markers of pathogenesis and target identification, and also if they change over time in ways that are more sensitive than the clinical outcome assessments, so that as long as we can correlate them with the clinical outcome assessments, we might be able to look at clinical trials in shorter periods of time because of them. So I'm going to start with some of the biomarkers that we're looking at. So this initial picture is just a picture of skin. And the reason I'm showing it is that there's lots of different cells in the skin, but there are some peripheral nerves in the skin, and they attach to things like the hair shaft. They attach to corpuscles that help you feel movement. And if you can find ways to actually look at changes in Schwann cells, then you can look at consequences of mutations genetically in CMT1B and CMT-X, both of which are caused by changes in Schwann cells. And we started this with CMT1A, which is why this is at the top of the slide. And this is with work that was really enabled by John Severin. And what John was able to do was he was able to identify 50 or 60 different genes that are only made by the myelinating Schwann cells and not made by other cells in the skin. And so he made little DNA tags for those. And then he linked them to tags like you might get at a grocery store and barcodes. These are colored tags, and each one is different for each gene. And you can then you perform a skin biopsy, and you can look at what genes are identified, which ones are upregulated, and which ones are downregulated, just in the myelinating Schwann cells from a single little dinky skin biopsy. And this is for CMT1A, and it just shows that you can detect the elevation of PMP22, which is the cause of this. But we have done that in CMT1B samples, and we've done it in CMTX samples, and we're working on putting together publications identifying those. In addition, you can look just at blood, and you can look for proteins that mark nerve damage. So even in CMT1B and CMTX, they're caused by problems in the myelin. Ultimately, like we talked about this morning, nerve axons degenerate. And when they degenerate, proteins that were in the axon get released into the blood. And the main one is called neurofilament light. And here is just showing on CMT1X and CMT1B, like the strongest marker we have so far is again for neurofilament light. So in other studies in mice, it's been shown that neurofilament light is elevated, and with successful treatment, it goes back to normal. There's a study in a genetic neuropathy in humans called familial amyloid polyneuropathy, in which neurofilament light is also elevated as axons degenerate, and it becomes normal with successful treatment. Therefore, we would expect and hope that for patients with CMTX and CMT1B, if treatments are successful, we would be able to use normalizing values coming back down here for neurofilament light as a biomarker for these diseases. Now I'm going to go into a little bit more molecular biology, so you have to bear with me here. That cells like Schwann cells or nerve cells or muscle cells, they use DNA to make messenger RNA, which then makes a protein. But there are other RNAs that they make that don't make proteins, some of which are called MicroRNAs. And what these do is they attach to parts of other genes and actually turn them down frequently. And these are called microRNAs. And if you look, you can look in blood and you can identify microRNAs that are released and that may correlate with the disease and may play a role in the disease process. And what we've done is we've identified a number of these microRNAs, and we've looked at them not just in CMT1A, but in CMT1B and CMTX. And this is a particular slide from work that John and I are doing on a microRNA called Myomir 206. So just to back up a bit to put this in perspective for what we're doing, we're looking in blood for protein and for RNA biomarkers of CMT1B and CMTX. We're looking at skin biopsies to look what happens to other genes made by the Schwann cells when there's a mutation in those. And we're also beginning to look at MRIs of the leg to look at for sensitive ways of disease progression. And this is an example from a patient with CMT1A, but as I'll show you, we're also doing this in patients with CMT1B. So here is the neuropathy scores for these three patients. One is very low, 5, that means mild. One's in the middle, 19, that's more severe. And here's one that's much more severe. And here is muscle. This is a bone up here. This is the calf. And you can see that this patient starts to get a little bit more white here. That's intramuscular fat that's accumulating, not because the person's out of shape, but because of cells in the muscle that change and become fat cells. And here is a more severely affected patient. And what you can do is you can color code each individual muscle there, and you can look at the changes from baseline to 1 year. And what we found is that in CMT1A, it's the most sensitive method of disease progression that we can use in a year's period of time. And as I'll show you, we're doing this in CMT1B and CMTX. So these are some of the studies that are being sponsored by the CMT1A, excuse me, by the CMTA through STAR that have to do with CMT1B and CMTX. And I'm going to focus on just some of these. So here is the biomarker project where we are looking at patients' biomarkers with CMT1B and 1X. And actually, that's the one I wanted to focus on this slide. I'm sorry, the slide got changed a little bit. Let me just go ahead one. So I'm going to back up here because I want to say some information that's not exactly on this slide. So what the CMT Association has done is that they've recognized that these biomarkers are important, the MRI part of it is important, and the clinical outcome assessments are important. But what we need to do is know how these work together. In other words, what changes in the MRI mean in terms of changes in the biomarkers in blood, in terms of what this means with the clinical outcome assessments. And this needs to be done with the new clinical outcome assessments that we've developed, like the FARM, the CMT Health Index, and the ones that have been developed since we had the CMT exam score. So they've sponsored what we call the 3-gene study, and this is a study of 5 sites, which include Iowa, London, Rochester, Milan, Italy, and one that I'm embarrassed to say I'm forgetting, University of Pennsylvania, in which we are all looking at patients with CMT1B and CMT1X, 12 at each site for a total of 60 patients. And we're doing all of these clinical outcome assessments, the blood and skin biomarkers, and the MRI studies on each of the patients. And we hope to have that data done within the next 2 or 3 years, which will be able to link all of these together for CMT1B and CMT1X to be clinical trial ready for these diseases. And again, this is just to put to show you a slide of the active partners working on CMT1B and CMTX through the STAR Alliance, which is extensive. So I hope I haven't been too confusing with that. And again, these are ongoing processes, but I think that hopefully you can appreciate that we're serious to this. We've recruited patients well for all of these studies. And again, these are ongoing. So I'm going to stop here and answer questions and turn it over to you, John, to host that.
Thank you, Dr. Shai. We only have one question in the Q&A. So if anyone else has any questions that you wanted more detail about, feel free to click on Q&A near the top and type it in. But the one question in there, Doc, is does weight or resistance training help? How might weight and resistance training help?
So I think that weight and resistance training does help, and I'm going to try to answer it in 2 ways. So putting on more weight puts more stress on your joints, which just makes everything harder. Resistance training can help up to a point. So if you do resistance training and you try to lift as much weights as you can, you're not going to be able to make atrophy go away. But if you do resistance training, you can help the nerves and their muscles that they're still connecting with work as well as possible. But it's important to remember that CMT1B and CMTX are length dependent. So problems are worse at the hands and at the ankles. So that if you start doing things like trying to maximize the weight you can bench press and you are using free weights, you run the risk that your upper arm muscles are going to be stronger than your wrists and fingers. And I fear for people dropping the bar on their chest and injuring themselves. So not trying to maximize the amount of weight you can lift, doing repetitions and making sure that you really are making your wrists and ankles sort of able to handle the weights. And I would advocate using machines rather than free weights.
I can back up your point from the patient perspective by showing you my 10-pound weight that I keep next to my desk chair. Mm-hmm. I don't try to push it with a 30-pound like I used to. I do more repetitions with 10 pounds, and I find that I have no pain from it, for me, speaking for myself. And that's how I get my strength, is by more repetitions of smaller amount of weight.
Okay. Right. And if you feel like a wreck the next day, then you're lifting too much weight. Right?
Amen.
Okay.
Amen. Next question. Is it helpful at this point to request a leg MRI to monitor one's progress?
It's not able to be done clinically yet, because to do this requires very precise measurements. They have to be done at a certain distance below the head of the tibia, for example, and it requires even little changes can sort of throw off the results and put noise into this, if I can use that term. And so the way these studies are done, in fact, is that all the institutions that are involved with them have to do sample control studies on people who don't have CMT. They have to be sent to a central institution where they're evaluated to make sure the protocols are reproducible from one site to the other. And again, all the analysis of these is done at a site in London. So ultimately, I think that this may be something that can be used clinically, but not currently. And also to look at the different muscles, like I showed you, that's a demanding task. So, I mean, if this wasn't a research study, this would be an extremely expensive evaluation.
Okay, appreciate that. How does someone get recruited for these studies?
So there are 5 sites, and if you're interested in being part of these, the sites, I'll say them again slowly and see if I can remember them all. So in the United States, it's the University of Iowa, it's the University of Rochester, it's the University of Pennsylvania. If you are in the United Kingdom, it's the University College London in London. And if you happen to be living in Italy, it's the University of Milan.
Awesome. I've also just put into the chat, everyone. No, that's a question and answer. Let me put this into the chat. There you go. Our link to the Patients as Partners form on the CMTA website, where you can fill that out with all of your information. And then if you're ever a good match for one of the research projects that we are funding and working on, you would be contacted directly. So that's another way to potentially get involved.
And we're recruiting for those studies, but unfortunately, I mean, we try to do these as it costs money to do these studies, and so we try to do this as efficiently as we can, but we use all the money for research, and we don't have funds for travel. So
let's see. Is there any connection of CMTX and circulation problems in the feet and legs?
So there's no direct cause and effect. People with CMTX, and really with all types of CMT, tend to lose muscle mass in their legs, and that makes it more difficult for muscles to pump blood up and down the legs. And so discoloration, like blue and reddish and cold feet, are from the CMT, but not because of circulation abnormalities.
Okay. What is CMTX6?
So that's pushing it for me. So I'm going to just, so CMTX, there are different genetic types, okay? And almost all of them, like way over 90% of them, are CMT1X caused by the mutation in connexin 32. There are some very rare forms of CMT caused by mutations on the X chromosome, and those go up at least through 5. So 6 has me a little bit stumped, so I'm not sure what that is. Okay.
So we're going to, for that last question in the chat, I've just posted here a link for your physical therapist to get everything from. I know we didn't answer every question, but the session is up. So if anyone wanted to ask their question that didn't get answered, feel free to send it to info@cmtausa.org. And Dr. Shai, thank you for a really informative session.
Well, thanks. Thank you very much.